We're from Telema High School, and today we have two exchange students with us. I'm from Thailand, and Eric's from Brazil. So we're doing research on sea urchin, and our research is called sunbathing sea urchins. So our test organism is purple sea urchins, or Strongylocentrus purpuratus, also known as the pincushion organism. They are invertebrates. Okay, so they live on rocky shores from California to Alaska.
Strongylocentrus purpuratus use their 2 feet to move their diet of brown-green algae and decayed matter to the mouth. Their predators are sea otters, sunflower stars, and California sheephead, which is a type of fish that's not pictured. A cool fact about them is they decorate themselves with shells, algae, and rocks to avoid predators and the sun's rays. So our research question is, how is a sea urchin's speed and algae coverage affected by different parts of the light spectrum?
Okay, so to go about making this happen, we had 3 different basins, and we did it indoors, and then we had used those basins again outdoors. And we had one that all of them were the same temperature, and then we had one that we covered with cardboard, and we covered it indoors, and then we covered it outdoors. Then we had one that was half exposed under the lamp, and then outdoors it was half in the shade and half in the sunlight. And then indoors we also did one that was fully under infrared, but outdoors that wasn't possible because of shade. And so we placed 1 urchin in each basin for 5 minutes, and then in the basin we had a bullseye. And we put some algae in the middle so that we could see how the urchin responded and how far they moved. And so we had 2 replicates, and we observed the speed of movement, which we calculated using the bullseye. So we would measure how far they had moved, and then we'd calculate their speed because we had them in there for 5 minutes. And the activities of spines, we did similar to the other group, where we would we observed 1, and then we called that 5, and then we would rate their activity on that scale from 1 to 10. And then we also eyeballed the amount of seaweed cover in a percentage, how much of their body was covered with seaweed. And so in our results, as you can see on the x-axis, we have the different light treatments. So there's 2 outdoors and then 3 indoors. And then on the y, we have the algae cover percentage. And so you can see that the sea urchins especially avoided the algae in the infrared light, and they're especially attracted to the algae when they were exposed to sunlight. And again, same setup. We've got the different light treatments on the x-axis, the 2 outdoors and the 3 indoors, and then on the y, you can see the speed, which is after 5 minutes, we calculated that. And so you can see that they were a lot slower when they were shaded and covered, when they were exposed to no light at all.
So the conclusion, their activity would increase in half shaded and half sunny because the ultraviolet light had some sort of effect on them. Their mental probably, so they try to get out of it. And then the environment, the more low tide means more light, so they try to hide themselves and camouflage from predators. Then with the infrared light, the changes were more in the temperature, so they got less activity. And then it's something that we can show with the global warming. So was proved correct from our hypothesis of if sea urchins are exposed to brighter light, then they will be more active and bury themselves in seaweed to hide from the light. And we learned that the infrared light had no effect on their interests to burrow under algae. If we could try to study it again, we would use a different species and add water temperature as a variable to see what effects prolonged infrared light might have on the organisms. I want to say thank you, Joe. Mr. Fong, Rachel, and sunburned sea urchins.
Any questions?
Any student questions?
Yes.
How do you calculate the say like algae or whatever? How do you figure that out? We had a bullseye that was 1 centimeter apart between each circle, and so then we looked at that and measured it and found the speed.
But you mean the algae coverage?
Yeah.
Well, they didn't really burrow under the algae. We just had it there in the middle, and then they would just move around. And some of them would like try to. You couldn't like blop it all together. It was just kind of spread out a little bit. And so some of them would really move towards it, and some of them wouldn't at all. Yes.
Do you think it would affect the movement of them if you use different colors of light instead of just infrared or like ultraviolet, like light, like different colors?
So
Yeah.
like a different part of the spectrum.
Yeah.
I think that they would react similar to how they reacted to infrared light. That these are just not used to infrared light because that's not what they get in the wild. They're used to ultraviolet. So I think they'd have a similar reaction if we used a different part of the light spectrum.
All right. No other student questions? Questions from the general?